Hello, welcome everyone to our online demonstration today. I am Olivier, Sales Manager at Inscoper, and I'm pleased to present you the new scanFRAP solution. I have a few slides to show you before meeting my colleague Julien at the microscope. During the whole presentation, feel free to write your questions in the chat. They will stay anonymous and we will answer them after the demo. So the scanFRAP is a turnkey solution for FRAP and all applications that require laser manipulation. You can see on this scheme, it consists of an optical module called laser illumination controller, an electronic box called device controller that controls the microscope and all its devices, and a software, I mean a graphical user interface, to set up the acquisition sequences and process the images. For those of you who are not familiar with the way Inscoper controls the microscope, in fact, the full acquisition sequence is run by this device controller independently from the computer. This innovative way of controlling the microscope allows full signal synchronization and removes all software latency, whatever the acquisition sequence. The scanFRAP is used for applications in live cell imaging, mainly to interact with specimens in the living state and observe their response to a laser action. So what are the advantages of this solution? The scanFRAP is a full software and hardware package. It means that this is not only about adding a new imaging technique on your microscope, but it is also replacing its current acquisition software with the automation and image acquisition solution from Inscoper. Users will benefit from a modern software environment to easily set up their multidimensional acquisition sequences, including or not photomanipulation techniques. This is of course interesting to upgrade or renew an aging microscope, but it is also a good solution to build and customize a new system with FRAP modalities. I would like to conclude this introduction by highlighting some technical features. The laser illumination controller uses galvanometric mirrors to direct the laser beam on the specimen. This is something quite conventional on the market. What we do differently is the resolution of the laser parameter controls. The illumination controller can manage 1,000 points at high frequency scanning with a very accurate spatial resolution for each point. It can also adjust the focus and the numerical aperture that are nice features for most demanding needs. We can, of course, talk more with each of you about your specific applications. So now it's time to move to the microscope for the demo with Julien.
Now let's move to the online demonstration. So I'm very happy today to present you the Inscoper scanFRAP. So let's open the Inscoper software. Okay, so now as you can see, all devices are initializing. We will create a new project. And now we are ready to work. So the Inscoper user interface is based on a user journey with 3 main steps. There is a configuration step, then an acquisition step, and finally a visualization step. So here we are in the configuration window. In the left part of the window, as you can see, we have some settings for the camera, including the exposure time and the binding. Here it will be the place for live image visualization. We will come back in this area later. And in the right part of the window, we have access to all motorized elements of the system. So for example, we can move objectives, we can also move the stage and the focus with these virtual joysticks. And we also have access to all settings of our light source. So here we have a lumen core light source, and we can select some channel individually. So we can make it manually, or when we made it once, we can save these settings, and here we can reload them. So for example, here, if I click in this configuration, as you can see, some settings will be loaded. So now, if I click on live, we are now able to see our cells. Okay. Okay. So here, as you can see, there is some parameters to modify the contrast and the threshold of our image. We also have access to here some settings for make crops. So for example, if I select these tools and I click on this button, I can make some crops on this image. So let's confirm. Okay, as you can see, we can see our nuclei here, here, and here. Okay. We also have access here to another button that can be used for change the LUT of our image. We have some, we can make some color, or some we have some presetted LUT here that can be used for acquisition. So here we do not need this option. Okay. As all devices, the Inscoper user interface can control the Inscoper scanFRAP. So here, as you can see, we have some settings that we can modify to adapt and optimize the scanFRAP to our biological experiments. For example, we can modify the density of each point that will be performed during the acquisition, and increase, for example, the density, by modulating the X spacing and the Y spacing. We can also modulate the pulse time. Before starting a FRAP experiment, we need to make a calibration. The scanFRAP have an automated calibration protocol, and to have access to this, we just have to click on this button. Before clicking to this button, we have to change the sample. For the calibration, it is more easy to make this calibration on a very fluorescent sample. So for that, we use this kind of slide. It is a fluorescent slide, and it will be easier for the calibration. So I will just move this on the microscope. Okay. Okay, it is good. And now I can click on this button. So here, as you can see, we are in the calibration window. We first have to select the camera we want to use and the channel we want to use. Here, for example, we only have one wavelength, but you can have several. Then you have to select if you want to reuse a calibration you previously done, or if you want to make a new one. So for this demonstration, let's make a new one. So we will move with this joystick these spots to each red cross. Okay, so it's good for the first one. Let's make it for the second one, the third one, and the last one. But it is the only manual step of the calibration. The next steps are fully automated, as you can see. So the scanFRAP head makes this pattern, and it is good for the calibration. So now I confirm, and now we can start to make some area to bleach. So for example, we can make some lines, and I make the fire preview. As you can see, we can see these forms. We can also make fill forms, including a circle like that, and we can exclude some area of this fill form to make this new area. And as you can see, there is a donut-like pattern that can be very useful in live cell biology. If, for example, we want to bleach the cytoplasm of a cell without affecting the nuclei. So, okay, all is good now, so I can stop the live, and we are able to go to the second step of the user journey. It is acquisition. So for that, let's move to our cells. So I have some cells that were transfected with fluorescent proteins. This protein will label the endoplasmic reticulum, And today we will make an acquisition of a FRAP-experiment. So we will bleach an RRI on these cells, and we will monitor the recovery of the fluorescence. Okay, so now we want to see our cells. So we'll go to this multi-channel dimension, and here we will have we'll select the good channel. So okay, I confirm, and here when I click in this button and I go on live, as you can see, we can see our cells. So this cell is very good. It is this one that we will use for all the experiments. Okay, so I stop the acquisition. So today we do not need focus. For this first step we only need the time dimension. So for that we will make, for example, 10 time points with an interval of 10 seconds, for example. I confirm. So it is good for the first sequence that we will call the pre-FRAP sequence. Okay. As you can see, we can modulate the order of the dimension for acquisition. So now we make the first sequence, so we will make the second one. In this second sequence, it will be the one in which we will make the FRAP. So for that, we do not need any dimension except the last one, it is photomanipulation. So now we will add an area to bleach. As you can see here, we can load a calibration channel. So it is this one we calibrated just before. We now have to select some RRI. So for this, I will go on the image and select, for example, this area. Okay, it is good. And now I just have to add this area here. So in this photomanipulation protocol, I will use this channel to FRAP this RRI. Okay, and now I have access to two parameters. So the pulse time, as you previously said. So for example, we will spend 50 microseconds per pixel, and we can select here the pulse iteration that correspond to the number of iterations that the laser will spend of each pixel. So here, for example, we want that the laser comes to each pixel 5 times. And the laser power is good at 100%. So I confirm the second sequence is perfect, and now I just have to make a third sequence. In this one we will monitor the recovery of the fluorescence. It's a post-FRAP sequence. In this sequence we will need to have more time points. So for example, this is good with 5 seconds of interval. Okay, so I enable all of this dimension we do not need today, and I add the same channel. Okay, so I confirm, and now all our sequence is fully ready to acquisition. So now I just want to show you this button. It is for data processing. It is here that we are able to adjust the way we will be able to see data during the acquisition. So here I selected the raw plus graphics. So we will be able to see live images with the graphics to monitor the recovery of fluorescence. So let's save this acquisition on the disk. Okay, and now let's start the acquisition. In fact, this acquisition was, I previously done this acquisition because it was quite long, so now I am able to show you it in fast forward. So now we can see in the left part our cells, and on the middle we can see the evolution of the fluorescence intensity. So for example, we can see now there is a FRAP that occurs. with a rapid decrease of the fluorescence intensity, and progressively we have a recovery of the fluorescence. Okay, so now the acquisition is finished, so we are able to see all images that were taken during the acquisition. So here we have the 3 sequences with all images that are available. We can have also here a play button to see again the sequence. Okay. We are also able to go here to see some graphics. So here in the ROI we can see the evolution of the fluorescence. Okay. And now if we want, we can export all this information in a .csv file, as you can see here. So I hope you have here all the information you wanted to know about the scanFRAP. And if you have, if you want to have more information or a personalized live demonstration, do not hesitate to contact us. I will be happy to make it for you.